Hi, hello, good morning. Today we are going to discuss about the cytology. The cytology is the study of individual cells, whereas the definition of the cytopathology is branch of pathology that deal with uh, manifestation of disease at the cellular level. Normal and the abnormal cells obtained from exfoliative imprint and scrape were observed under microscope and recorded throughout the 19th century. In 1861, a drawing of a keratinized squamous cell carcinoma was published, and this was drawn from a pharyngeal secretion obtained from a postmortem of a patient who died of a growth in his throat, even though it was not identified as keratinized squamous cell carcinoma in at that time. Today, by looking at the drawing, one can identify the cause of the death. After 22 years of this incident, George Papanicolaou, the father of the cytopathology, was born, and the famous Papanicolaou smear and the Pap stain was named after him. Today, cytology includes fine needle aspiration, is considered and recognized as the cost-effective, simple, accurate, and a safe procedure in diagnosing and taking treatment decisions for certain pathological conditions. So, when we consider the cell, so the cell can be considered as the unit of life. The word cell was first described by Robert Hooke. In Latin, the word cella means a little room. For the study of cytology, the cell is broadly classified into four parts: nucleus, cytoplasm, cytoplasmic membrane, organelle. Examples of the organelle: mitochondria, uh, Golgi apparatus, ribosomes, lysosomes, etc. So, when we consider cytological features of atypical cells, the features of abnormal or the atypical cells in cytology preparation, which are helpful in the diagnosis of malignance. So here in this table describing the features, right, of the atypical cells. When we consider the component of the cell, so here we are discussing about normal cell and the abnormal cells. So when we consider the cells number, first one number, cell number, normal cells, it is low because normally what happen when the cell is growing, initially it is immature and it is growing up and matured, and after the lifespan it will apoptosis and it will die and it is shaded out from the area where it is grown. So what happens? Shaded cell only in normally if you observe any specimen in any area, you can see the very low number of cell because cells after finishing their lifespan, it will shade it out from the area. So very few numbers we can see. But in the abnormal cases, what happens? The cell numbers are increased because here apoptosis is lower but it's abnormally growing and released from the area where it is formed, right? When we consider the arrangement of the cells, so in normal cells, cells are organized, monolayered, tight clusters. So when we consider normal cells, it normally what happen? It's organized, right? Uh, There is a that means the well organized. When we consider the one cell and another cell, they are similar, little bit almost similar. And when we consider this is monolayer because it is shaded after lifespan, it is shaded out. So it will, the layer by layer, it will remove from the area where it is formed. And other thing, it is tightly clustered. First layer, it will finish the lifespan, it will remove from the area, so we can see the specimen. But in the case of abnormal uh, condition, so the cells are disorganized because there is no any similarity organization among the cells, and sometimes we can see scattered the and other things, sometimes we can see the single cell, right? And other thing, we can see thick sheet like structures. So when we consider the here we are talking the cells, right? About the cells. First one is the number, other one is the cell arrangement. Then next one is the cell variation on size and the shape. This is we call pleomorphism. So usually in normal cells we can see absence of cell size variation and the shape variation. But in abnormal conditions, we can see the various size, the shape of the cyst. Then, when we consider the nuclear and cytoplasmic ratio, in normal condition it is low, right? But in abnormal conditions, the nucleus and the cytoplasmic ratio, normally we call NC ratio. N mean the first letter of the nucleus, C mean first letter of the cytoplasm. So NC ratio. So what happened in the normal condition? In here, the normal condition, what happened? Matured cells are really shaded out from the cells, the cluster. During that time, what happened? The matures, the nucleus and the cytoplasm are matured, and the nucleus are compact. So that's why the nucleus size is smaller. But when we consider the mature cells of the cytoplasm, it is larger, volume is larger. So that's why nucleus is very smaller and the cytoplasm area is higher. So compare with the ratio, the low. But in the case of abnormal cells, in here, normally what happened? We can see immature cells. So in during maturity, what happened? The nucleus becoming a larger nucleus when we consider immature stages. In here, the cytoplasm and nucleus and cytoplasm ratio is higher because the cells are immature cells are active by active cells. Actually, they can divide. They can involve in uh, mitosis. So the nuclear contents are active and spread like that. So what happened? The nuclear area is higher and the cytoplasmic area is low. So NC ratio is higher. During the maturity, what happened? The nucleus, it will, the nuclear chromatin, it will condense. So it's supercoiled and it appears smaller. right? But during the maturity of the cytoplasm, during the maturity, nucleus, both nucleus and the cytoplasm are matured. So during that time, nucleus size is lower, becoming lower in mature cells. From immature to mature, the nucleus size is decreased, but in cytoplasm size is increased. So what happened in C ratio is lower in normal cells, but in abnormal condition, what happened? We can see in mostly we can see, most commonly we can see immature cells. So the NC ratio is higher. So here there are some examples for you. Here you can see first picture left side the cancer cells and the normal cells. Here you can see well organized normal cells. You can see boundary, the cell cytoplasmic boundary, nuclear boundaries, and there is a shape. We can shade. This is round, oval, like that. But in cancer cells, what happened? The membrane is irregular, but in these normal cells, you can see the membranes of nucleus or the cytoplasm. They are evenly distributed. But in the case of cells, cancer cells, it is irregular in when we consider nuclear membrane and the cytoplasmic membrane. And other thing inside the cytoplasm, we can see some extra uh, ingredients, right? So in this picture, first one left side, cancer cells, a dark pinkish color. At center is the nucleus. Surrounding area is the cytoplasm. But when we consider normal cell, yellow color, at center it is nucleus. It is evenly organized shells, and we can very nicely observe the shape of the nucleus, the cytoplasm. So they here the comparison between healthy cells and the cancer cells. So here when we consider healthy cells, normal cells exhibit a large cytoplasm. So we already discussed during the maturity, the nucleus sizes decrease and the cytoplasm sizes increase. That's why the larger cytoplasm. When we consider the cell individually, this is spherical shape of nucleus. So inside we can see spherical shape nucleus. Uh, but uh, shape of this healthy cells standardized in shape. So when we compare two cells, so yes, these are uh, almost similar, right? Standardized cells. So we can say this cell is for example, if we consider squamous cells, we can say these features are the if we observe this is squamous cells, right? But intact tumor suppressor genes and apoptosis gene. So here this is controlled by that suppressor gene and the apoptosis gene. When we consider cancer cells, it is exhibit a smaller cytoplasm because the these are cancer cells are immature cells, so nuclear is immature. It is larger, right? So the cytoplasm content is low. Non-spherical shape of the nucleus because during the cancer formation, the nuclear membrane is uh, default. So the actually we cannot see spherical shape, right? Another thing inside the nucleus we can see multiple nucleoli because during the mitosis or mitosis, we can see the nucleoli. Active cell it contain that means dividing cell. It's contain nucleoli. So here we can see multiple. That means it's dividing cell active, right? Non-standardized in shape. So here we cannot say the shape. When we compare two cells, the size, shape, all may vary. And damaged tumor, suppressor genes and apoptosis gene. Here it's involved uh, suppressor genes, apoptosis genes, and damaged tumors. So when we consider the variation in size and the shape, so here you can see normal and the cancer cells. So normal cell in larger variability shape in nuclei. For here first picture you can see normal and abnormal cells. In cancer cells you can see larger variable nucleus, but in normal cells pinkish color, very small nucleus, round in shape, right? Another one, many dividing cells, disorganized arrangement. So here you can see second picture. So here normal cells you can see the nucleus size can observe it is similar. When we consider the cells, it is the cytoplasm. It is almost similar. That looks like same, and it is organized very well. That's near cluster like, but in here cancer cells it is single. There is no organizations, but here in the cancer cell second picture you can see uh, the nuclear, the cytoplasm area, and some here you can see uh, uh, nucleus is only we can see. But third picture, variation in size and shape. So here the consider the normal cell, okay, normal, but in the cancer cells you can see in the nuclear verification size and shape. When we consider a single cells, the size and shapes are vary. Fourth picture you can see loss of normal features. So normal and normal cell fourth mm, picture, you can see normal cells. There is a nucleus, cytoplasm, nuclear boundary, cytoplasmic boundary. But in this case, the features of the cells we cannot uh, say the cell features, right? But normal cells we can see this is squamous cell, this is parabasal cell, this is intermediate cell. But in the case of cancer cells, their normal features of the cells is lost. Then when we consider nucleus cytoplasmic ratio, if the cells maturing, the ratio NC ratio is lower, right? So here the example, if the immature, very initial cells, this is very immature cells in that last picture. Consider the last picture. Or otherwise, first we will discuss first one. First picture, the NC ratio is 0.1 because the nucleus is smaller, cytoplasm is larger. So when we consider first and second pictures, actually compare with first one and second one is somewhat immature. Like that, second and third, again third is immature compare with second. Like that it's gone down, and last picture it's indicate the very initial stage of the cell, right? So in that situation nucleus are very higher, occupy very high area of the cells. The cytoplasm is very smaller because it is very active. So and it will mature during the maturity. uh, so cytoplasm increase, volume is increased, the nuclear volume is decreased. So here you can see the during the maturity what happened? Not only cancer cells, normal cells also, they when they become matured, so it will happen, and right. So normally here these pictures describe the myeloblastic, right, myelocytes maturation. The left side first picture, what happened, this is the myeloblast. This is actually a very initial cell, immature cell. So in here, the almost all the area occupied by the nucleus. The cytoplasm is look like a dream-like structure. So it when it is this is here in here, NC ratio is very higher. So it is the next stage of the uh, myeloplasm, right? Blast series. The one is a myelocyte. So myeloblast become a myelocyte. During that time, what happened? The nuclear content is reduced and the cytoplasm is increased, right? So again, these myelocytes it become a metamyelocytes, right? Left side, second picture. So in here also, the ratio is reduced, and the shape also, it's the shape of the nucleus also varying. Then it's mature into the uh, myelos, myelocytes, right? In during that time, what happened? Then cytoplasm is increased and the nucleus is decreased. Right here, another comparison of normal and cancer cells. So normally, cytoplasm, normal cell cytoplasm is larger. We can see in single nucleus, and the single nucleolus also we can observe. When we consider the chromatin, fine chromatin evenly distributed. So, but in cancer cells, cytoplasm is smaller because these cells are immature, and we can see in multiple nuclei and multiple and large nucleoli we can see, and coarse chromatin. Coarse chromatin means in some area chromatins are thicker, some area no. So that's why we call coarse. Fine mean evenly distributed inside the nucleus. But in the case of cancer cells, the chromatins are not evenly distributed. Some area it appears very thicker, some area no, some area little bit like that. So in other thing, pleomorphism, yes, cell pleomorphism means size and shape variation, and we can observe in cancer cells. Same time, high NC ratio and irregular chromatin. That's why we call irregular chromatin, right? Okay. So NC ratio high due to the nucleus, right? Regular chromatin mean the arrangement of the chromatin, right? So next one, we previously we discussed about the cell, overall cell. Now we are going to discuss about the nucleus changes, right? Nuclear differentiation between normal and abnormal conditions. So when we consider the nucleus, it is the size first, the smaller, and abnormal condition enlarged. We already discussed, right? So this picture describe very well here. This one can see first picture. The nucleus, you know, the smaller, but in here cancer cells it is larger. And nuclear arrangement ordinary. So here you can see another one, disordered, piled up and crowded. So here nuclear arrangement orderly. There is a order in normal cells, but abnormal cells, this is not in order, disorder, uh, filled up and crowded. Then when we consider nuclear membrane, so in normal cells we can see regular, but in abnormal cells we already discussed irregular and thick. When we consider color of the nucleus here, uh, hypochromasia, normally we call the hypochromasia. Normal cells we cannot see in the hypochromasia, the color hypochromasia increase the colors basophilia, right? Why it is increased? Because in abnormal cells the chromatins are not compact, right? Loosely arranged, so that's why it's getting the hypochromasia. When we consider the chromatin in normal cells, fine and regular, but in abnormal cells, coarse and irregular. When we consider the nucleoli, so here one, smaller one, normal cells, sometimes it will not appear, right? Mm, we cannot see. But in abnormal cells, we can see larger, prominent, irregular. Sometimes we can see multiple nucleoli. So mitotic feature, that's mitotic features figures mean uh, dividing features. So in normal cells, what happen? Usually few because already it is matured ones, right? But in abnormal cells, yes, it is increase and abnormal. So here you can see the compare the normal and abnormal cells. So normal cells we have nucleus, especially here not only nucleus, cytoplasm and the nucleus, nucleoli. Though here normal cell larger cytoplasm. So when we consider the nucleus spherical shape of the nucleus, chromatin fine arrangement. That's why the color is even. Wherever seen in the nucleus, it is same color. Then nucleolus single, right? If it is one is present, we call nucleolus singular. But if more than two is present, nucleoli, right? So cancer cells, the cytoplasm is smaller because uh, the nucleus occupy larger space, and the regular shape of the nucleus shape is irregular, and the cell boundary, nuclear boundary also irregular, and you can see multiple nucleoli because it has mitotic features. That's why it's contained nucleoli, where we can see multiple nucleoli during the cell division. So it is dividing features. That's why it's a higher number in nucleoli. Another thing is coarse chromatins because it is the distribution of the chromatin is varied. That's why coarse chromatin features. So when we consider the size of the cells in normal and abnormal cells, so here normally how we can calculate nuclear volume over cell volume, right? So here for example, uh, here 300 nuclear volume, 1200 cell volume. So 300 over 1200, 1 of 4, nearly this is 25 percentage normal cells. But in the case of abnormal, nuclear volume is higher, cell volume is low, or sometimes same. What happened? It's increased, right? So second figure you can see normal healthy cells, cytoplasm, nucleus. There is a shape, size, definitive organization, definitive shape. But atypical cells, what happened? Irregular size, shape, and the color of the nucleus, hypochromasia, and the predominant nucleolus, right? Sometimes in matured cells we cannot see in the nucleoli, but it is present, but we cannot see. But normally in abnormal cells we can see prominently. right? Other one is the nuclear arrangement in normal and abnormal cells. Here, normal branchial cells here, first figure you can see the arrangement. You can see all the cells same similar place. The cells are nucleus are arranged. But in here, so figure second pictures B and C, here you can see the arrangement. Nuclear arrangement it may vary. Similar cell, first one is the normal cell, second and third are carcinoma cancer cells. So the look at this, compare the first figure and the second and third. So arrangement of nucleus it's vary, right? And the shape, consider the shape, size. The third picture you can see the nucleus, the color, the transparency inside, right? You can see the prominent nucleoli. So nuclear membrane in normal and abnormal. Here first figure normal cells, but in uh, other pictures you can see the more changes. Nuclear membrane, right? It is vary. So the last picture, high grade breast cancer, you can see the membrane, shape of the membrane, right? Some places it's folded inside, right? Like that you can see in some the picture B, papillary thyroid carcinoma, you can see inside the nucleus you can see some abnormal features. We will discuss later what is that, right? Now you should understand the differences between the normal and abnormal cells. So nuclear membrane, again here very nice picture. So here you can see the right side one is normal one, the left side is abnormal one. So here you can see the nuclear shape, right side, there is a boundary, right? And you can see nucleoli, the nuclear chromatin evenly distributed any area, they're same. But in abnormal, irregular nuclear contents here, the some area is folded, goes the folded inside, and the chromatin cause chromatin. The other thing, the nucleus is enlarged, darker nuclei compare with normal one. Then you can see in the prominent nucleoli, right? Then hypochromasia. Here you can see the different color, dark color. Hyper mean higher color is higher, right? Abnormal cells. The chromatin you can see here. The in normal cells, chromatins are contents, right? But in abnormal cell, it is loose chromatin, so that's why it active and it is easily inverse from one cell to adjacent cells. Right, next one is nuclear chromatin pattern and radial distribution. Here you can see the cells, fine granular, medium inside the chromatin. You can see we already discussed fine the coarse chromatin. So here you can see coarse chromatin. What is coarse? In the first picture A and B, you can see the differences. And second, right side topper one, there are four type of cells. Fine granular means finely granular in spreaded all the area. But B, second picture, it is medium granular, right? Granules are compare with first picture. Second, there is some granules. But third one is coarse granules. So we can see in some area dark spot like that. In some cases, in some abnormal cells, we can see very peripheral chromatin. The chromatin membrane, it's contain the chromatin. So this is peripheral chromatin. So this is the abnormalities of the nucleochromatin pattern and the distribution. So when we consider the nucleoli, normal cell and abnormal cells, Normal cells contain nucleoli, but we cannot see because chromatins are compact, so darker in color. But in the case of abnormal cells, we can see multiple nucleoli because it is loosely arranged. Chromatins are loosely arranged, so we can very clearly we can see in immature cells. So what happened? How the cancer cell is formed? No, initially it is no normal cells, right? Normal lifespan. After if it is mature, lifespan is it reached, so it will the gene suppressor gene and other things it's involved on the that mature cells and it apoptosis happen and it will destroy. So in some cases, some co- normal cells exposed to some conditions, so it will change genes. So, so mutation we call. So during that mutation, the cells nucleus are automatically dividing and increase the cell number. So that's we call cancer. So what are the stimulus increase the multiplication. So some we when we give the input stimuli, so it will automatically normal cell become abnormal cells. So sometimes growing signal, cellular stress, so DNA damage, hypoxia, drug treatment, energy demand status, active rDNA repeats, P53 pathway, cell cycle. These are the input stimuli. So when it happen to the normal cell, so nuclear will response and cell grow, stress response, DNA damage repair, telomeres, uh, genomes regulation, cytoplasm biogenetics, P53 pathway and cell cycle. These also involve and these normal cells become abnormal. So what happened in here? Actually, nuclear dysregulation is happen. So what happened? It is supporting the proliferative signal. So normal cancer cells. So it will normally increase the cells, the number of cells, and resist to cell death. So now the mutation is happen due to the input stimuli. So what happened? It is automatically resistant to cell death. So automatically it will grow, right, and increase the number of cells. So that's why we can see abnormal cells also in our smear. And inducing angiogenesis, blood supply increase. Other one avoiding the cell growth suppressor. So he what happened? Suppression of the cell growth is reduced. So automatically it's growing, right? The activating the invasive and metastasis. So during this activation, what happened? It will automatically uh, move to other part of the adjacent cells or far away from mm, the origin. Uh, Enabling replicates immobility. So here, uh, motility means sometimes after it will grow, right? For very initially, it will grow and it will make immature, intermediate cell. It become a mature cell, and after that, apoptosis happening. It will remove from the cells, but it will prevented during the cell cancer. So when we consider the cytoplasm of the cell, normal and abnormal, here cell boundary already preserved. So we know the whatever nuclear cell boundary, cytoplasmic boundary and nuclear boundary, it is preserved. But in abnormal cell, it is lost. But when we consider the cytoplasmic stain, it is normal in normal cells, but in cytoplasmic stain, these are abnormal stains. When we consider the background of the cells, it is normal, but abnormal cells, it is abnormal backgrounds, right? And other thing, when we consider the necrosis, we can study what is necrosis and other thing. So normally, in normal cells, it is absent, but in abnormal cells, abnormal condition, we can see the necrosis and the hemorrhage. So here we can see hemorrhagic in abnormal condition, but in not in a normal condition. So here is the background of the uh, things. For first one is the first figure left side. It is clear background, but in second picture, the right side one, it is something mm, dirty, right? Not clear. It is overlapping on the cells and we can clearly demonstrate the cell. But in first figure, yes, very nicely we can define the cells, nucleus and other things. But this is over the second figure overlapping the background, right? So uh, demonstration is difficult. But here is the hemorrhage. The A and B. A is hemorrhagic smear. B is the clear one. So we can very clearly differentiate or demonstrate single cells. But here hemorrhage blood smears, so it is overlapping the cell. Actually, we cannot demonstrate the cells, individual cells morphology. So this is the necrosis. So here the first left side figure A, B, C, D, you can see the necrosis. Normally alive cells, this is one. Apoptosis, during the apoptosis cell, all cell, when we consider the single cell, it will uh, form vesicles and vacuoles and it's destroyed. But in necrosis not like that. So we will learn and general pathology, what is necrosis and other thing. We can see uh, necroptosis and at final it will come necrosis, right? The here the left right side figure you can see viable cells, primary necrosis. What happened? Uh, C is the early apoptosis. Normally inside we can see in vacuoles and other things, lysosome formation. This is natural process, apoptosis. So what happened? Inside the cell, if lifespan is finished, what happened? Automatically cell uh, produce acting the genes, suppressor gene, and it will act on this and it will produce lysosome and toxic substances and it destroy the cells. But in necrosis also like a apoptosis, but it is not natural things. It's due to some abnormal condition, right? Like a dead cells we can see, but not not all death, but it is actually due to uh, abnormal conditions, right? So here you can see normal and abnormal cells, normal cluster of normal cell, intermediate cell. So here when we consider single cell, cell by cell, yes, we can see the similarities, organization, right? But in abnormal cells, not like that. The cells to cell, they can vary, right? So relatively large amount of cytoplasm in normal cells, clear nucleus stain, fine chromatin granules in the nucleus, the smaller nucleolus, and the smooth nuclear border. A stain artifact in one cell where another smaller white blood cell overlap, right? But in cancer cells, larger size of the nucleus compared to the total cell size and the dark stain of the nucleus because the nuclear chromatin is coarsely arranged. The larger size of the chromatin clump in the nucleus. So we can see nuclear clumps. Fourth one, the larger nucleolus and irregular nuclear boundary. These are the features. By using these features, we can differentiate normal and abnormal cells. So here you can see normal and abnormal dividing cells, right? Here normal cells, you can see the surrounding. At the center, mm, we can see the immature cells, right? Uh, so next class we will go to discuss about the urine cytology. Thank you for your attention. Please subscribe our channel, like our channel, and share.